In March 2026, a team of geneticists ran the most advanced DNA analysis ever conducted on the Shroud of Turin. They used next-generation sequencing technology, the same tools used to decode ancient genomes from 50,000-year-old Neanderthal bones. They extracted genetic material from samples that had been sitting in storage since 1978, when scientists first gained official access to the cloth. And they expected to find what you would expect to find on any ancient fabric that has been touched by human hands for 2,000 years. Human DNA. They found that. But they also found something else entirely. Something that had no business being on a piece of linen. Something that forced the research team to write sentences into their published paper that read more like the setup to a mystery novel than a scientific report. Because buried inside the fibers of the world's most studied relic were microorganisms so specialized, so geographically specific, so dependent on a particular extreme environment that exists in only one place on Earth, that their presence on this cloth becomes one more piece of a puzzle that the scientific community has been trying to solve for nearly 50 years. This is not the DNA you think they found. This is the DNA nobody was expecting. And once you understand what it means and where it can only come from, you will realize that the Shroud of Turin stopped being a religious debate a long time ago. It became a scientific one, and the science is not going the way the skeptics predicted. Let's start from the beginning of this particular investigation, because context is everything when you're dealing with evidence that is thousands of years old and has passed through the hands of hundreds of people across dozens of countries. The study published in March 2026 was led by Gianni Barcaccia, a professor of genetics and genomics at the University of Padua in Italy, together with an international team of researchers. The study was titled simply DNA Traces on the Shroud of Turin, Metagenomics of the 1978 Official Sample Collection. The samples they analyzed came from the first major scientific examination of the cloth, conducted in October 1978, when a team of over 40 American scientists, physicists, chemists, biologists, imaging specialists, forensic experts, were given rare and unprecedented access to the Shroud in Turin. Those scientists spent 5 days conducting non-destructive testing on the cloth. They also removed small samples, dust particles, linen fibers, microscopic fragments, using sticky tape pressed to different areas of the cloth corresponding to the face, hands, back, and feet of the body image. Those samples were cataloged and stored. And decades later, the 2026 research team got their hands on them and ran metagenomics, the most powerful form of genetic analysis available, on what had been collected. Metagenomics is not your standard DNA test. When you sequence a single person's genome, you are reading the genetic code of one organism. Metagenomics sequences everything simultaneously, every fragment of biological material in a sample, regardless of what organism it came from. Human DNA, bacterial DNA, fungal DNA, plant DNA, animal DNA, viral DNA, the DNA of organisms that have no common name, because most people have never heard of them. Metagenomics reads it all at once, and then researchers use databases and algorithms to identify what each fragment belongs to. It is essentially a complete biological inventory of a sample. And when you run that analysis on a piece of ancient linen that has existed in the world for potentially 2,000 years, passing through countless environments and countless human hands, you are reading the entire biological history of everywhere that cloth has ever been, every environment it was stored in, every person who ever touched it, every organism it ever came into contact with. The 2026 study was effectively a complete biological autobiography of the Shroud of Turin, written in DNA. The human genetic findings were extraordinary and have been widely reported, and we will get to those. But the findings that stopped researchers in their tracks, the findings that generated the most careful and cautious language in the paper, were not the human DNA. They were the archaea. Archaea are not bacteria. They are not viruses. They are not fungi. Archaea belong to a completely separate domain of life, one of the three fundamental branches into which all living things are divided: bacteria, archaea, and eukarya. Archaea are ancient microorganisms that diverged from the common ancestor of all life on Earth billions of years ago and evolved into forms capable of surviving in environments that would kill virtually every other living thing. The archaea found in the 2026 Shroud study belong specifically to the class Halobacteria, a name that does not mean they are bacteria, but that they are archaea that thrive in conditions of extreme salinity. Halophiles, salt lovers. These are organisms that have adapted over billions of years of evolution to survive in environments where the salt concentration is so high that it would cause the cells of any ordinary organism to collapse and die. They thrive in places like salt lakes, salt mines, hypersaline coastal pools, and most specifically in environments like the Dead Sea. The Dead Sea is one of the saltiest bodies of water on Earth. Its salinity is roughly 10 times that of the ocean. The water is so dense with dissolved minerals that fish cannot survive in it. Most plants cannot grow near it. Most bacteria cannot live in it. But halobacteria, the specific class of halophilic archaea found on the Shroud not only survive there, but thrive there. The Dead Sea region is one of the most consistently documented environments on Earth for this particular class of organism. And the 2026 study found them in the fibers of the Shroud in sufficient quantities and with sufficient genomic integrity that the researchers noted their presence was not incidental. The halobacteria found on the Shroud showed what the paper described as a pronounced overrepresentation of osmotically resilient species, organisms specifically adapted to high salinity, within the archaeal community reconstructed from the samples. This was not a random contamination event. The sheer dominance of salt-adapted archaea within the archaeal fraction of the Shroud's microbiome indicates sustained exposure to a high salinity environment, or storage under saline conditions, for a significant period of time. The Vatican News Service, which is not given to sensationalism, commented directly on this finding and stated plainly that the presence of microorganisms thriving in extremely saline environments, such as the Dead Sea, was confirmed. The researchers wrote that their findings suggest the Shroud resided in the Middle East, in a saline environment consistent with conditions found near the Dead Sea, adding what they described as a new piece of evidence to the body of scientific material supporting the relic's authenticity. Let that sit for a moment. A cloth that a 1988 radiocarbon test said was made in medieval France is carrying extremophile microorganisms so specific in their environmental requirements that their presence points, in the words of the scientists who found them, to the region of the Dead Sea, not to France, not to Italy, not to England, where one of the 3 1988 dating labs was located, to the Dead Sea. But the archaea are only one category of unexpected biological material found in this study. The full picture of what the 2026 team discovered reveals a cloth that has lived a life of extraordinary geographic breadth, a life that does not fit the biography of a piece of linen manufactured somewhere in medieval Europe and then kept in France and Italy for 700 years. The researchers also found DNA from Mediterranean red coral, Corallium rubrum. This is a specific species of coral endemic to the Mediterranean Sea. It grows on rocky underwater surfaces throughout the region and has been harvested since ancient times for jewelry, religious objects, and decorative carvings. Finding its DNA on the Shroud means the cloth came into contact at some point with objects made from Mediterranean red coral, which is consistent with the historical record of rosaries, crucifixes, and reliquaries made from this material being placed in physical contact with the Shroud over centuries. But it is also consistent with the cloth, having spent time in Mediterranean environments. The geographic DNA footprint of this cloth is pointing overwhelmingly to the ancient Mediterranean and Middle Eastern world. Then there's the extraordinary diversity of plant DNA. The 2026 study identified genetic material from at least 19 different plant species, including carrot, red wheat, corn, bananas, and peanuts. Some researchers have pointed to this plant diversity as evidence of contamination, and they are right to note it. Corn, bananas, and peanuts are all New World crops that did not exist in the ancient Middle East or medieval Europe, meaning their presence on the Shroud reflects contact with the world after the Columbian Exchange of the late 15th century, when European explorers began bringing American crops back across the Atlantic. The researchers themselves noted this explicitly, writing that the significant environmental contamination of the Shroud likely occurred in recent centuries, particularly following the voyages of Marco Polo and Christopher Columbus. This is important because it demonstrates exactly what metagenomics can and cannot tell us. The plant DNA from American crops speaks only to handling and storage in the modern era, not to where the cloth originated. The halophilic archaea are a different matter entirely, because they cannot be explained by contamination from curators or pilgrims touching the cloth in Turin. No casual human contact deposits extremophile salt-loving archaea in quantities sufficient to dominate the archaeal fraction of a microbiome. Their presence requires direct environmental exposure to a high salinity context. The animal DNA found in the study adds another layer to the biological portrait. Dogs and cats together accounted for approximately 44% of the animal DNA detected, the result of pets in storage environments over centuries. Cattle, pigs, chickens, goats, sheep, horses, deer, and rabbits were also detected. These are the animals of the ancient Mediterranean and Middle Eastern world. They are also the animals of European domestic life across the medieval period. Their presence cannot be used to pinpoint geography in the way that the halophilic archaea can, but they are consistent with a cloth that has moved through the world of ancient livestock markets, temple environments, domestic storage, and cross-cultural handling over a very long span of time. Now let's talk about the human DNA, because what the 2026 study found in the human genetic fraction of the samples is where the findings from this paper collide most powerfully with the earlier 2015 study led by the same researcher, Johnny Barcaccia, and where the full picture starts to become something no simple forgery hypothesis can accommodate. The 2026 study identified multiple human mitochondrial DNA lineages on the shroud. Mitochondrial DNA is passed from mother to child and is used by geneticists to trace maternal ancestry and population origin. Several lineages were identified. One, called K1A1B1A, matched the mitogenome of the 1978 official collector, confirming that at least part of the human DNA signal came from the person who physically handled the samples during collection. This is important because it shows the methodology was sensitive enough to detect even single contact contamination, which means it was also sensitive enough to provide meaningful signal from the cloth itself. Beyond the collector's lineage, the researchers identified H1b, which is common in Western Eurasia, and H33, which is prevalent in the Near East and is frequent specifically among the Druze population of Lebanon and Syria, a community whose genetic profile connects directly to ancient Levantine populations, including Palestinians and Syrians. Now add the findings from the 2015 Barkachia study, which used different samples and different methodology but analyzed the same cloth. That earlier study identified over 55% of human DNA on the shroud tracing to Near Eastern lineages, approximately 38 to 39% tracing to Indian genetic lineages, and less than 6% to Europe. The Vatican noted this distribution and offered an explanation that is historically grounded. The presence of Indian DNA can be explained by the documented use of fine Indian linen at the Temple of Jerusalem, referenced in ancient rabbinical texts as hindoyin, used for the garments of the high priest during the afternoon rituals of Yom Kippur. The word shroud itself derives from the Greek sindon, meaning fine linen, which may be etymologically connected to Sind, the region of the ancient Indus Valley civilization internationally celebrated in antiquity for producing the finest quality woven linen in the known world. The cloth's name, its DNA, and its fabric origin may all point to the same place. This convergence of evidence, Near Eastern human DNA, Indian textile trade genetics, Dead Sea extremophil archaea, Mediterranean coral, 1st century linen origin confirmed by X-ray cellulose dating, Jerusalem pollen embedded in the weave, is not the biological profile of a piece of cloth that was forged in 14th century France. Forgeries do not contain halophilic archaea, calibrated to the Dead Sea region. Forgers do not source their linen from the ancient Indus Valley trade routes that supplied the Temple of Jerusalem. Forgers do not leave a geographic DNA signature that points, from 5 different independent lines of biological evidence, to the ancient Middle East rather than medieval Europe. The 2026 team also used the metagenomics data to make a specific discovery about the cloth's repair history that deserves separate attention. Within the samples collected from the shroud's reliquary, the container in which the cloth is housed, the researchers found 2 distinct textile residuals that were radiocarbon dated independently of the main cloth. These threads dated to periods between 1451 and 1622 CE, and between 1642 and 1800 CE, respectively. These date ranges align precisely with 2 well-documented historical events: the fire of 1532 in Chambéry, France, in which the shroud was nearly destroyed and was subsequently repaired by poor Clare nuns who patched the fire-damaged areas, and a further conservation effort in 1694. The dating of the repair threads is internally consistent with the historical record. This matters enormously because one of the most credible critiques of the 1988 radiocarbon dating, which assigned the main cloth to between 1260 and 1390 CE, is that the sample used by all 3 laboratories came from this same lateral edge, the section most likely to have been contaminated by the medieval repair materials. The 2026 study did not date the main cloth again, but it did provide independent radiocarbon confirmation that repair materials from the 1534 and 1694 interventions are present in the fabric of the relic. The 1988 test may have been dating a patch. And then there is the image, because everything discussed so far is about the biological history of the cloth, where it has been, who has touched it, what environments it has experienced. The image is a separate category of mystery entirely, and the 2026 DNA study does nothing to explain it. No DNA analysis can, because the image on the shroud is not a biological phenomenon, it is a physical one, and what physics has established about it over the last 50 years is, in its own way, more startling than anything the DNA has revealed. The image on the shroud, as established by the 1978 Sturpey investigation and confirmed by every subsequent analysis, exists only in the outermost layer of the linen fibers, a depth of approximately 200 nanometers, which is thinner than a single-cell wall. It cannot be reproduced by any contact process because contact processes transfer material, and there is no transferred material on the shroud. It cannot be reproduced by heat because heated cloth fluoresces under ultraviolet light, and the image does not fluoresce, even though the 1532 fire scorch marks on the same cloth do. It cannot be reproduced by any known chemical process because chemical reactions penetrate deeper into the fiber than 200 nanometers. It cannot be reproduced by photography because photographs do not contain three-dimensional spatial data, and the Shroud image does. It encodes accurate depth information about the body that formed it, which is only visible when the image is processed through computer software designed to read topographic relief. In June 2025, a study published in the International Journal of Archaeology by chemical engineer Thomas McAvoy of the University of Maryland applied principal component analysis and pattern recognition algorithms to high-resolution photographs of the Shroud and confirmed what the 1976 VP8 analysis first showed, that depth cues are intrinsic to the cloth's image rather than artifacts of photography or artistic technique. The image knows how far away the body was from the cloth's surface at every point. It carries that information in the intensity of its coloring. And only one known physical mechanism can produce that kind of uniform, surface-only, depth-encoded discoloration on a linen fiber. A burst of vacuum ultraviolet radiation emitted from the surface of the body uniformly in all directions, at an intensity of billions of watts, lasting for approximately 1/40,000,000th of a second. Researchers at the Italian National Agency for New Technologies, ENIA, spent years attempting to replicate the image formation characteristics, using state-of-the-art Excimer lasers generating vacuum ultraviolet light. They succeeded in producing coloration limited to the uppermost fiber surface, matching the spectral profile of the Shroud image, matching the depth of coloration, matching the hue. But the scale required to produce the same effect across a full human body image was beyond any conceivable technology. Their calculation, the energy required to form the image on the Shroud, spread across the full surface area corresponding to a human body, would require approximately 14,000 Excimer lasers operating simultaneously. The most powerful UV laser facility on Earth cannot approach this energy level. Paolo Di Lazzaro, who led the ENIA research, stated clearly that their experiments show the image properties are produced by a burst of photons in a very narrow range of parameters, and that while this does not prove the image was produced by a laser, it does demonstrate that vacuum ultraviolet radiation is the only known physical mechanism capable of producing these specific characteristics. No natural phenomenon known to science produces this kind of radiation from a human body. There is no known example in the history of human science of a dead human body producing an image of itself on a piece of cloth. No chemical process, no biological process, no artistic technique, and no known natural physical process explains it. The image is there. It has been studied by experts in physics, chemistry, optics, spectroscopy, and materials science for half a century. They have not explained it. They have only confirmed that it cannot be a forgery because no forger in any century, medieval or modern, has demonstrated the ability to produce it. So what does the 2026 DNA studies actually tell us in the end? It tells us that the biological record of the Shroud of Turin is not consistent with a cloth manufactured in medieval Europe. The human genetic profiles on the cloth point to the Near East and ancient Indian trade routes. The plant pollen embedded in the weave points to Jerusalem. The cellulose of the linen has aged 2,000 years by X-ray analysis. The flax was grown in the Western Levant. The blood is real, type AB, with elevated trauma biomarkers. And now, from the microbiome reconstructed by the most advanced sequencing technology available in 2026, the cloth carries the biological fingerprint of an organism that can only tell one story about where this linen has spent time: the edge of the Dead Sea. None of this is human. Not in the sense that any human being put these archaea on this cloth deliberately. They grew there. They were deposited there by an environment. And the environment that deposits halobacteria in these quantities, and in this specific class of osmotically resilient, salt-adapted life, is the environment of ancient Judea, the salt-saturated landscape surrounding one of the most historically significant bodies of water in the world. The DNA on this cloth is speaking. It is not speaking in a human language. It is speaking in the language of ancient microbes that have been living in the same salt-soaked landscape for millions of years. And what they are saying, to anyone willing to listen to the data, is that this cloth was there. Not in France, not in Italy. There, in the desert, by the Salt Sea. In the land where, 2,000 years ago, a man was crucified, wrapped in linen and laid in a tomb. The biology does not prove what happened in that tomb, but it confirms, layer by layer, study by study, molecule by molecule, that the cloth's origins are ancient, Middle Eastern, and exactly where Christian tradition has always said they were.